We usually think about genetics like it's some kind of digital code, right? A's, T's, C's, and G's. But what if it's also a physical machine? Today, we're going to dig into something wild. How the simple act of twisting DNA can actually send messages that decide the fate of a whole population. Let's get into it.
So, where does the story start? Well, it starts right inside you, in your gut. It starts on your skin.
They're literally everywhere. Bacteria are a constant part of our world. We live with them every single second. But this super familiar fact, it hides a really deep mystery about how they actually work. And that mystery brings us to this question. What if you had a bunch of bacteria with the exact same genes, but they acted totally differently? How is that even possible? That's the puzzle we're going to try and solve. Okay, so to get our heads around this puzzle, let's look at a pretty cool lab experiment. Scientists took some bacteria and gave them two new genes. Simple enough, right? One gene makes a red protein, the other makes a green one. But what they saw happen next, well, it was not what they expected at all. Okay, just look at this. It's wild. You have three different populations of bacteria, and they all have the exact same red and green genes. But look at what they do. The first group, the cells make a choice. They're either red or green, a total binary switch. The second group, almost all red. And the third, it's a mix, but they're definitely leaning green. I mean, come on, same genetic code, three totally different results. What is going on here? All right, the answer starts somewhere you might not expect. It's not in the genetic code itself. It's with a physical problem, a huge physical problem that every single bacterium has to solve. Basically, a massive storage crisis. So think about this. A typical E. coli bacterium is about 2 microns long. I mean, it's microscopic. But if you took its DNA and stretched it all the way out, it would be thousands of times longer than the cell itself. That's just mind-boggling. Yeah, we are talking about 4 million base pairs of genetic information. All of that has to be crammed into this tiny, tiny space. It's kind of like trying to stuff 26 miles of thread into a single pill capsule. So how on earth do they pull it off? Well, here's the cool part. The solution to this crazy packing problem, it's also the key to our whole genetic puzzle. And the secret, it turns out, is all in a simple physical act, twisting. The official name for this is supercoiling, and it's basically what it sounds like. You take DNA, which is already a twisted double helix, and you twist and fold it back on itself over and over again until it becomes unbelievably compact. Probably the best way to picture this is with something like a garden hose or even an old phone cord. Imagine holding both ends and just starting to twist one of them. You can feel that tension building up, right? Well, if you keep twisting, what happens? The hose will spontaneously just knot up and fold over on itself. It's trying to relieve all that stress. That's exactly what supercoiling is. So the DNA goes from this kind of relaxed, open loop to this super tightly wound, spaghetti-like mess. It's a brilliant way to pack everything in. But, and this is a big but, the story does not end there. Okay, now we get to the really crucial connection. This whole physical twisting thing, it's not just about storage. It's actually a form of communication, a way for genes to talk to each other. Yeah, researchers in this field basically say that twisting is a shockingly effective way to send a signal from one place to another. And it makes sense, right? Think about it. If someone twists your arm, you get that message instantly. There's no ambiguity. Well, DNA is using that exact same principle. So here's how it works. For a cell to actually read a gene, its machinery has to pull the two strands of the DNA helix apart, just for a moment. Now imagine doing that to a twisted rope. If you untwist one little section, all that twist, all that tension, it has to go somewhere, right? What happens is it creates an over-twisted tight knot of tension ahead of the gene and an under-twisted loosey-goosey section behind it. So get this. The simple act of reading one gene sends these waves of physical tension rippling down the DNA strand. And this is the whole shebang right here. The physical arrangement of the genes, how they're oriented on the DNA strand, matters a lot. If two genes are pointed towards each other, their twisting forces literally smash into each other, creating this super high-tension zone. If they point away from each other, those forces just kind of spread out and disappear. And if they're lined up in the same direction, one gene's twist can kick off a chain reaction in the next one. It's a mechanical language written in pure physical force. Okay, so now we have all of the pieces of the puzzle. Let's go all the way back to the beginning back to those weird bacterial colonies and see how this mechanical language explains everything. And here it is, the solution. Remember that colony where the cells had to choose red or green? Well, their genes were in that convergent, head-to-head, high-tension arrangement. When one gene turned on, the physical force it created would literally shut the other one off. It was a my way or the highway situation. And the colony that was mostly red, that was the divergent arrangement. The one that favored green, you guessed it, tandem. It wasn't magic. It was pure mechanics. And this is so much more than just a cool lab trick. This physical communication system is something bacteria use all the time to make actual life or death decisions out in the real world, including inside of us. Yeah, one of the most important examples has to do with something called biofilms. Biofilms are these tough, slimy, resilient colonies of bacteria. You've definitely encountered them. It's the plaque on your teeth. But they can also be way more dangerous, like the stubborn infections that can grow on medical devices. So, nasty pathogens like Staphylococcus aureus use this exact twisting mechanism for a deadly divide-and-conquer strategy. They use that same convergent, high-tension gene setup we saw before, and what does it do? It forces the colony to split up the work. Some of the bacteria focus on growing and spreading the infection as fast as possible. The others focus on building that tough protective biofilm. It's the exact same red or green choice, but in the real world, it's grow or defend. And that simple mechanical switch is what can make an infection so incredibly hard to get rid of. When you think about it, this discovery really changes how we see genetics. It's not just a static digital code, a string of letters. It's a dynamic, physical machine operating on principles of tension and force. And that leaves us with a pretty mind-blowing question to chew on. If a simple twist can communicate so much information, what other physical forces are hiding in our DNA, shaping life in ways we're only just starting to scratch the surface of?